Welcome, everyone. Thank you so much for joining us today. My name is Shannon Holmes. I'm a medical physicist here at Standard Imaging, and I would love to talk with you today about our scintillation detectors for when those millimeters matter most. Precision, precision, precision. I guess it is your time. Be precise. We're here to support you in the work that you do, and we are happy to help you out in the best ways that we can. So just a little housekeeping before I get started. I am recording the webinar today, so if you have to step out in the middle or if you'd like to share it with a colleague afterwards, keep an eye on your email. Later today or tomorrow, you should be receiving a link to the recording of the webinar. If you have questions as I'm going through things today, please enter them in the question box in the GoToMeeting dialog. I'll try and address those as they come up. But if I miss one, I will circle around back to that at the end and get to your questions. If you don't know a lot about us, Standard Imaging is based in Middleton, Wisconsin. We're just outside of Madison. It's absolutely beautiful here. All the flowers are blooming. We were founded in 1989 and started with a small portfolio of products, but have grown significantly since then to designing and manufacturing a full suite of QA products for radiation therapy. So if you have questions about whether we have other types of QA devices available, the answer is probably yes. Please contact us. We'd be happy to tell you about it. So scintillators in particular, why would we go down this route with scintillators? They're not the usual detectors that people are often used to. But as we're getting down into these very, very small fields where that volume averaging and the precision placement of your device is so critical, there are definitely some advantages to organic scintillators. The first is that you've got water equivalents for MV photons and electrons. And the real key to this is that as your field size approaches the size of your detector, the perturbation of the detector itself plays a larger and larger role in your measurement. And so if you have a detector that's not perturbing your beam right there at the point of measurement in these small fields, you can be a lot more confident in the accuracy of your measurement. They can be made very small. We haven't seen dose or dose rate dependence with these, which is an advantage as well when we're looking at FFF beams, for instance. They're generally energy independent. The beam quality conversion factor, again, because of that water equivalence and not perturbing the beam and not showing that difference between energies, beam quality correction factor is unity, which is a huge load off your plate if you've looked at the small field correction factors for other devices. They do show a minimal temperature dependence. It's about 0.1% per degree. This doesn't really matter just for QA measurements, but we do like to put it out there if people are looking into experimental things where you're maybe looking at different temperatures. There is a slight temperature dependence. We don't usually correct for it, but if your calibration measurements are significantly different than your actual measurements, you may need to consider that. The other nice thing about these fibers is the fiber itself has no metal parts in it whatsoever that would perturb an MR beam. So we do make a longer fiber for use with MR Linacs so that you can keep the electronics, the optical detection outside the 5 Gauss line, but otherwise they have been successfully used with MR Linacs as well. So what does our configuration look like? I mentioned we have an organic scintillating material, and so with depending which fiber you've got, the W1 was our first generation scintillator. The W2 is the more recent generation, and there's both a 1x3 and 1x1 millimeter option for the W2. They're 1 millimeter diameter, and then the difference is the length of the scintillator here. So 3 millimeters long for the W1, 3 or 1 for the W2 fibers. Ionizing radiation interacts with that scintillator to make optical light, and so we have this coupled to an optical transfer fiber. The difficulty with this, and part of the reason that it wasn't commercialized until just recently, I guess somewhat recently, the past 10 years, is that the electrons irradiating that optical transfer fiber create Cherenkov light. And so this acts as basically a stem effect, a contamination that we have to remove from the measurement. The method that we use is a 2-channel chromatic correction based on a publication by Matthew Guyot et al. I list the publication here. I'm happy to point you to that on the MedPhys site if you need as well. But with that method, it definitely enabled us to make this, this commercial device. The other thing about scintillation detectors is that they're not absolute dosimeters. They're not things like ion chambers that have that can be sent to a standards lab for calibration, and you expect that that value holds for 2 years. These devices are plastic, clear plastic, and if you've seen acrylic phantoms that have been irradiated, you can see that they start to yellow. And because we're looking at the color transfer through these fibers, that change in color of the fiber over time decreases the signal. And so they do need a periodic recalibration as it accumulates dose. You lose about 2% of the signal for every kilogray of irradiation that you give to it, just because of that degradation of the plastic. So they do need to be replaced periodically as well, as you get up to really high doses to your fiber. But something to be aware of, that they're not an absolute dosimeter. You do have to give them a known dose if you want the output in terms of dose, and then recalibrate periodically. A little more about that 2-channel chromatic correction. We have a blue scintillator, and so the majority of your signal is in this blue region. Cherenkov light is a broad spectrum contaminant, and so what we've done is we've split this signal into a blue region and a green region. We have you do a set of characterization measurements where the key here is that you're keeping the dose to the scintillator itself constant and just changing the amount of optical fiber in the field. Sorry about the telephone in the background. I'm not sure I said that. So what we have you do is what we call a minimum fiber configuration. This drawing is from the slab that we sell with the W1 that can be used in a virtual water stack in order to do your characterizations that way. The W2 was designed more for use in a water tank, so we have a smaller jig that's used in the water tank for the same purpose. And essentially what you would do is have your, for minimum fiber configuration, you'd have the fiber coming straight out of the field in either of these configurations, or excuse me, either of these holders. For the maximum fiber configuration, you would wrap the fiber around through this channel or through the channel here in in the water tank. And the idea really is, if you keep the dose to the scintillator consistent and change the amount of fiber in the field, then only your Cherenkov contribution is changing. And from those two measurements, from the difference in the signal in each of these two channels, we can calculate what's called a Cherenkov light ratio, or CLR. So difference in the blue channel between the maximum and the minimum configuration divided by the difference between the green channel for the same thing. Then for subsequent measurements, your scintillation signal alone is equivalent to that blue channel minus the Cherenkov, which is calculated by the green signal times the CLR. And that has been shown to be quite stable. If you're doing a dose calibration, that essentially becomes a scaling factor to this, just to convert from the charge that's collected from the photodiodes for collecting that optical light into a dose value. So you would give it, after doing the CLR measurements, you would give it a known dose and a reference field size, and be able to get your output in terms of dose at that point. This jig was designed so that you could use it with CyberKnife, with the 6 centimeter field of the CyberKnife kind of centered over the fiber here. If you're using a linear accelerator, you would actually probably be better off using a 10 by 10 field that covers the whole circle. The fiber, or the tip of the fiber, as long as you're not doing your dose calibration, does not need to be centered in your field. It just needs to get that consistent dose each time. So it's fine for the CLR measurements if it's offset within the field. You would just want to make sure you shift it back to the center before you do your dose calibration. So I mentioned that the W1 was our first generation device. It has been on the market since 2014. It has that 1 millimeter diameter, 3 millimeter long active area. There are a lot of publications available for it. One of my favorites is the AAPM and IAEA TRS 483, which is the guidance for small field measurements. It has tables for KQ factors for a number of detectors. The W1 is the only one in that entire publication that shows values of unity across every table that it's in, all the way down to the smallest fields. You'll note that some of the detectors don't show values for small fields, and that's because the uncertainty in the measurement was too high for that group to feel that they could list recommended correction factors for those smallest fields. The disadvantage of the W1 is it really was designed just to be a single point measurement system. So for, say, just output factor type measurements. It also does require a 2-channel electrometer, such as our Supermax, in order to be able to perform that Cherenkov characterization and the subtraction. The Supermax has a routine built into it so that it'll walk you through those characterization measurements. It'll save your calibration value and your CLR so that when you make measurements with that scintillator, you put it in scintillator mode and it will automatically do those calculations for you to give you your Cherenkov corrected output. If you were using someone else's 2-channel electrometer, you can still do those measurements, but all of the calculations would need to be done by hand because nobody else has that routine built in currently, as far as I know. The W2 was the more recently released one. It's our 2nd generation scintillator. It has its own dedicated electronics. And so this one, instead of having the fiber fixed to the optical detection system, which then has to connect to the 2-channel electrometer, we've got the optics and the signal processing electronics all within a dedicated electrometer. So that meant we could have a user replaceable fiber. It's just the fiber and a standard optical plug. So if your resident rolls the cart over top of the fiber and breaks it, or it just gets old, it gets too much dose, it's very easy for you to replace that fiber on your own. We also send it with 2 different fibers, a W2 1x1 and a W2 1x3. And the reason for that is really there are some times when you definitely need the 1x1 for the resolution for those extremely small fields. But there are also times when the 1x3 is advantageous in the slightly smaller, or excuse me, slightly larger world of small fields. Because it has 3 times the signal, and because that exactly matches the W1. So if you're comparing to W1 publications, that gives you a direct apples to apples comparison on the size of your detector. So the dedicated electronics here are called the Max SD for scintillation detector. You can do your point measurements, your characterization measurements, and individual point measurements directly with the Max SD. It has a web-based interface that you can control it from outside the bunker. The Max SD needs to sit about 1 meter away from the field that you're measuring because of scatter to those components does give you a little more noise in your measurement. But it's quite easy to use at that in that configuration. If you want to use it in your water tank, there is also a conversion from the Cherenkov corrected signal with your scintillator to an analog output via triax cable from the back of the Max SD that can be sent to your water tank. So from the water tank perspective, it looks like you're scanning with something like a diode. You don't apply bias to it, but when the beam's on, you get a signal from that detector. So you can use it for your small field scanning with whichever water tank you own. Here are a couple examples of just what that setup looks like. We have an adapter sleeve that goes on the scintillator. This extends that kind of stiffer stem of the scintillator out far enough that you're away from your scanning components. And it also makes it the standard diameter that you might have for your scanning chamber, so it fits with your scanning chamber brackets without you trying to clamp down super hard on that optical fiber. You could do your CLR measurements in your water tank with this configuration with a rectangular field that you rotate with your collimator if you don't want to use the blue jig that I show here. But these are that minimum configuration, maximum configuration looking down at the water tank, and then what we would use for scanning. Then you hook your Max SD up to your scanning electrometer, just like you would any other detector that you're scanning with. You do have to scan kind of slowly with the scintillator. It is a small signal device, and so it is a little more susceptible to the noise that just of your full electronic system. We do apply a gain. We amplify the signal that we're sending from the Max SD to your water tank electrometer. That really is just to make sure that we're well within the range that your water tank electrometer can measure well. It can't generally get down to these small signals we're getting from the scintillator without bumping into your electronic noise floor. So that's there. But we do recommend that you do about a 1 second measurement at each measurement point in order to get a good quality measurement curve. When you do that, here's an example of a publication from Paulina Galavis and the group at NYU Langone, comparing the W2 with film measurements. So you can see excellent agreement here on a 1-by-1 field. They also looked at dose rate dependence and dose dependence just to verify that there wasn't a change from the W1. We didn't expect any. It's the same fiber. It just happens to have the option for the smaller piece of scintillation material at the end of the fiber. But it's nice to have that confirmed anyway. Another interesting publication from, I guess it's a couple years ago now, 2019, came out of Richard Popple's group at University of Alabama at Birmingham, and they were looking at variant hyperarc targets, targeting lesions down to about 3 millimeter diameter, and trying to do some QA of that. So they were comparing the W2 with radiochromic film again for this. You can see images of the phantom plug that was used, I think in a lung phantom. But we also include a localization marker fiber for the W2 fibers so that you can actually see it on the images as you're trying to align. Because as you can see, when you have the W2 in the image, it's water equivalent. It's very hard to pick out either for planning or for localization where that active scintillation region is. So that's what I had for you today. I'm happy to answer questions if anyone has any. Otherwise, there's information on our website at standardimaging.com, or please feel free to reach out to your sales representative at sales@standardimaging.com. And I appreciate your attention today. Thank you so much for your time, and we look forward to talking to you more in the future. I'm not seeing any questions come in, so it must have been absolutely perfectly crystal clear, which either that or everybody's overwhelmed. But I'm going to go ahead and close the session here. Please reach out to us if you have any questions. We would love to talk to you more about it. Or if you have questions about any other QA equipment, we're here to help you. Thank you so much for your time today, everyone.